Share the screen again. There we go. All right, so what we're doing today is chapter 13. Yeah, we're a little bit ahead of schedule, but that's all right. I don't think we'll finish chapter 13, but I want to get close to finishing, and then we'll almost be a day ahead. And then I can get the review on our last lecture day, and then we won't even have a last lab. But if I don't get the review in the last lecture, we will have it in the last lab, which will be Thursday. What is that? About March 11th? Whatever that Thursday is. Any questions about what we're doing today? In the lab, there is no lab this week, so I will be there from 6:30 to 6:45. So if you have a question, come in from 6:30 to 6:45. If there are no questions at 6:45, I will log off. But if there are questions, I will continue until all the questions are answered. All right, so we were talking about viruses, and I believe I was on this slide the last time I was talking. Is that correct?
Yeah.
Thank you. So the multiplication of animal viruses follows a similar pattern to the multiplication of bacterial viruses that we call bacteriophages, but there are some differences. And I mentioned the only one major difference you need to know is animal viruses can multiply in the nucleus, whereas they can also multiply in the cytoplasm. And bacterial viruses only multiply in the cytoplasm. I already did that one. The multiplication of animal viruses do follow a little different pattern, and one difference is that their attachment to the cell membrane it differs because bacterial viruses attach to the cell wall of bacteria. Animal cells, of course, don't have a cell wall, so the virus attaches to the cell membrane. And they get into the cell differently. There is no mechanism for the animal virus to, I don't know, puncture, get through the cell membrane. They get through the cell membrane by endocytosis or by fusion. This picture is showing you endocytosis, and earlier editions of our textbook called it pinocytosis. And if you happen to have an earlier edition of the textbook, you should make a note that this is not correct. It's by endocytosis, where the virus attaches to the cell membrane, and then the cell membrane invaginates and invaginates, and then invaginates extremely, and then this will break off and come inside the cell. Sorry, I'm not trying to be moving that. Break off and come inside the cell, and the membrane will form around the vesicle, which will then move into the cell, and the membrane will reform up here too. And there you can see it there. And this is an actual, I think, electron microscopic image of that happening, although it's actually more than one virus. It's just that. And the same with this here. The other way that they can get into the cell is by fusion, and that is a virus that has a envelope. That envelope is a lipid bilayer, and it's phospholipids. And the lipid bilayer of the envelope can fuse to the cell membrane. And then when they fuse, let's pretend this was an envelope virus, and then it fuses. The envelope of the virus then becomes the envelope of the cell membrane, and then that's not shown correctly, so let's go to the next one. Right here, they can fuse together, and then the cell membrane just reforms, and then the virus is dumped inside the cell. Is that clear? How the two fuse together, and I don't know if I can show it with my hands or not, but they come together and then they touch, and then they fuse, and they just become one, and then the virus is dumped into the cell. There's a few other differences. I'm not going to talk about them. One difference is with the multiplication of our animal viruses, and that is bacterial viruses only multiply by the lytic life cycle and by the lysogenic life cycle. Animal viruses can multiply by the lytic life cycle if there is a lytic animal virus. Animal viruses do not have the lysogenic life cycle. Instead, they have a budding life cycle. And these viruses, an animal virus, instead of lysing the cell, which a lytic animal virus can do, and that ruptures the cell envelope, the budding virus, when it's finished, it'll come up to the membrane of the cell. And this can be the cell membrane, the nuclear membrane, or a membrane of the endoplasmic reticulum. So animal viruses that replicate by the budding life cycle come up to a membrane, and it can be the cell membrane, which is probably most viruses, but it could be the nuclear membrane, and it could be the membrane of the endoplasmic reticulum. And it just pushes on that membrane and buds it out, and then it rips off a chunk of the membrane, and that membrane reforms. So this right here rips off, and this part of the cell membrane joins with that part of the cell membrane, and the membrane reforms. And then a piece of the membrane is taken by the virus, and that becomes the envelope of the virus. And that is the budding life cycle for animal viruses. And here you can actually see a virus budding out of the membrane. I'm not sure, I guess that's the cell membrane, it's hard to tell. Any question about any of that? Let me blow this up just a little bit. You'll notice that in this membrane here, here's a regular part of the membrane. It's yellow. And then right here, you'll notice that there's blue things in the membrane. Those are viral proteins that the virus has transcribed and translated, and then the viral proteins goes into the membrane. And you'll notice that the virus will bud out of the membrane at a region of the membrane that has the viral proteins in it. That's just something you should know, but I'm probably not going to question you on that. But the point is that the virus puts proteins in the membrane, and then it'll bud out of that membrane. These proteins actually can serve as antigens so that the host can attack this cell because it has proteins on it that the host immune response can recognize as foreign. So this host cell could be attacked because of these viral proteins. But the proteins are needed by the virus. And these proteins could be the spikes of an envelope virus. Let's go way back. Remember, we're on slide 32. I'm not finding it. Oh, there it is here. So those proteins could be the spikes. Oops, too far. of the envelope virus. All right, any question about the budding life cycle? If not, let's go on. I should mention that in the budding life cycle, the virus usually does not kill the cell. So that this cell here, that is shedding budding viruses, and this cell here too, I don't know if that's another cell or the same cell, but maybe this cell here and that cell there. Generally, the budding virus does not kill the cell. That the virus is replicated at low enough burden that the cell sheds them slowly in budding, and the budding doesn't take enough of the membrane to kill the cell. So usually the budding virus does not kill the cell. If this cell dies, it's from the host immune system recognizing that it is infected with the virus and then mounting an immune response on that cell, killing it to try and get rid of the virus. Any question about any of that? All right. As we've discussed, viruses have different genomes. They can be double-stranded DNA viruses, single-stranded DNA viruses, double-stranded RNA viruses, and then two types of single-stranded RNA viruses, the plus RNA single strand and then the minus single strand RNA viruses. And they have different genomes, viruses, and each of these different genomes needs to have a slightly different mechanism for multiplying and undergoing biosynthesis of the viral genes in order for the virus to replicate. Any question about that? And we'll get back to that subject. We're going to talk about the DNA viruses first. Let me see if I can blow this up. Yeah, that's better. DNA viruses, don't memorize the table, don't know the names. We will talk about a few of them. We've already talked about the herpes viridae. The pox viridae is smallpox and cowpox, so we have talked a little bit about that. We haven't talked about the others. We will talk about adenoviridae, the adenoviruses. They cause human colds and a few other things. And I think that's about it. Anyways, there are 1, 2, 3, I think he's 7, 4, 5, 6, 7 family of DNA viruses that cause human diseases. And these are the diseases over here. You do not need to memorize this table. I'm just showing you that there are 7 families of DNA viruses that cause human diseases. And let's start talking about the DNA containing animal viruses. For DNA animal viruses like the adenoviruses and the herpes family of viruses, the DNA is generally copied in the nucleus. So DNA animal viruses normally replicate in the nucleus. And that's a rule of thumb that you can just memorize. Any question about that? The DNA, when it's copied, usually uses viral enzymes, but it can use host cell enzymes to copy the DNA. And the capsids and the proteins of these DNA-containing viruses, those are synthesized in the cytoplasm using the host cell machinery, meaning that's where a eukaryotic cell makes its proteins, and the virus uses that same machinery to make its proteins. Any question about that?
No.
All right, so once the viral DNA enters the host nucleus, one of two things can happen. The first is the viral DNA can be expressed The cell can produce new virions. Let's move on to the next slide. The DNA will be replicated because we're talking only about DNA viruses, and that's true if it's double-stranded or single-stranded. The DNA will be transcribed, making viral messenger RNA. The viral messenger RNA will be translated, making viral capsid proteins. The capsid proteins will get together with the viral DNA, the replicated viral DNA, making new progeny virions, and then the cell will shed these virions. And that will usually happen by one of two life cycles. We'll talk a little bit more about them later, meaning it could be the lytic life cycle or it could be the budding life cycle. Animal viruses do have one more, but it's close enough to the budding life cycle. So we'll just say those two for the moment. And that's the normal way that many DNA viruses replicate. But another thing can happen when the DNA enters the cell. The DNA can integrate into the host chromosome. So it can recombine with the host cell's DNA, and then the chromosome, or at least one of the chromosomes, will have a chunk of viral DNA in it. That we don't call the prophage because, of course, animal cells don't have phages, bacterial phages, because those are bacterial viruses. So they give it a different name. They call it a provirus. Very similar, but we call it a provirus. And that is a host cell that in the host DNA, there is a chunk of viral DNA integrated into it. Now, something that's different with this integrated provirus compared to the prophage of a bacteria cell is the provirus will permanently remain a piece of DNA in that host cell's DNA. It will never be excised. And that provirus will be replicated only when the host cell replicates its DNA. Is that true? Let me think about that. While it is a... no, that's not true either. Let me reword that. I didn't quite word that right. The provirus replicates when the host DNA is replicated. Okay? When the virus is multiplying, making new viruses, that the provirus DNA integrated into the host cell's DNA can also replicate. So when I said it only replicated when the host cell DNA replicates, that's not right, because this virus can replicate. I cannot, for the life of me, remember a DNA virus that does that. Oh, the papillomaviruses do. There we go. A papillomavirus integrates into the host cell's DNA, and while it's inactive, the virus isn't replicating. The provirus will only replicate when the host cell's DNA replicates. But while the virus is replicating, like the person is shedding papillomavirus, then the provirus part of the chromosome, meaning the viral DNA, can replicate. We already talked about this slide, so let's move on. This slide is showing you the multiplication of a DNA virus in more detail. The DNA virus attaches to the cell membrane of the cell and gets inside the cell. The vesicle is degraded, releasing the virus in the cell. And there's different mechanisms of degrading the virus. We're not going to get into that. It can be viral mechanisms or host cell mechanisms. But the important part is the virus is disassembled, and then the viral DNA gets into the cell and that viral DNA moves from the cytoplasm into the nucleus. And then while it's in the nucleus, the viral DNA will be transcribed into viral RNA, including viral messenger RNA. The messenger RNA will move into the cytoplasm. The viral messenger RNA will be translated into viral proteins. In the meantime, in the nucleus, the viral DNA will be replicated, and then the viral DNA will get together with a viral protein to reassemble the new virion, and then the new virion will leave the cell either by budding, which is not shown, or by the lytic life cycle, by lysing the cell, and then it will release not one virus, but lots of viruses in the lytic life cycle. The budding life cycle, the cell will release a few viruses will be budded. Any question about multiplication of a DNA virus that is an animal DNA virus? Everyone good with DNA animal viruses? If not, let's move on to the RNA animal viruses. There are, I believe, 15 families of RNA viruses, which I'm not even showing you all of them, that we need to talk about. And we don't need to memorize this table. I'm just showing you the animal viruses. Let me count them, make sure I got it right. 15 animal viruses that cause disease in humans. And these are the diseases here. We will talk about a few of these. The Picornaviridae has what do you call it? It's the family that produces the most human colds. So some of the picornaviruses cause human colds, and those are called mostly rhinoviruses. Hepatitis A is in this one. I was trying to remember. I thought there was another one in that. I think polio is in the Picornaviridae. We won't talk about that one. The herpes viruses, we've talked about herpes viridae. I don't think we talk about these. Coronaviridae, we're going to talk about that one, about 3 or 4 slides. Retroviridae, we'll talk about this one. The retroviruses are an RNA family of viruses, but they are an exception, and we'll explain why well, when we get to the retroviruses at the end of the RNA viruses. All of these other RNA viruses, we'll talk a little bit about the orthomyxoviridae viruses. These are the viruses that cause the flu, and then all the others we won't talk about. So don't memorize this table. I'm just trying to show you there's a large number of family of RNA viruses that cause human diseases. And we're just not interested in RNA families that don't cause human disease. The RNA containing animal viruses, here's 3 of them here. You'll notice they're different shapes and sizes. The multiplication of the RNA viruses, essentially their multiplication is similar to the DNA viruses. There are some differences though, because they have RNA as their genome instead of DNA. For a rule of thumb, for the RNA viruses, they replicate in the cytoplasm. So animal RNA viruses generally, as a rule of thumb, replicate in the cytoplasm. There is one exception, and you need to know about it because we'll talk about it, and that is the retroviruses are an RNA virus, but they do not replicate in the cytoplasm. they replicate in the nucleus. So the rule of thumb works for all RNA viruses except for the retroviruses. We're going to look at examples of the RNA viruses. The double-stranded RNA virus, how it replicates is really easy, and that is it has two strands, one positive strand, one negative strand. To replicate the positive strand, the virus just uses the negative strand and then uses it as a template to make the positive strand. I suppose I should be having it that way, but too hard to do. And then to replicate the other strand, it uses the corollary strand as the template. Any question about that? For the positive single-strand RNA viruses, how they replicate is the positive strand, single-strand RNA animal viruses replicate by making a negative strand. Up. Let's go ahead and talk about, there we go. Here it is. So the positive strand, single-strand RNA virus makes a negative corollary strand RNA and then uses this as a template to make more positive single-strand RNA viral genomes. Of course, the positive strand can be used by the ribosome, so we can transcribe this strand to make the capsid proteins in translation. And then the capsid proteins get together with the genome of the virus to make the new progeny virions. Any question about any of that? Now, for the negative single-strand RNA animal viruses, they replicate their genome by making a positive strand, single-strand RNA, that's the corollary to the negative strand, and then making the genome, the negative single-strand RNA, from the corollary, the positive strand. And only the positive single-strand RNA is transcribed, read by the ribosome to make the capsid proteins. And then the capsid proteins get together with the genome, the negative single-strand RNA virus, to make the new progeny virions. Any question about how animal RNA viruses replicate? So this slide is showing you really everything at once. We're looking at DNA. DNA, well, I guess it's not showing you the DNA being replicated, but it can be transcribed as messenger RNA. The messenger RNA can be translated into viral proteins. If the genome of the RNA virus is positive strand, it makes the negative strand, the antisense, and then uses that strand to make more of the positive strand RNA for the genome. And then the positive strand, single strand, can be translated and used as messenger RNA to make the viral proteins. And then the negative strand, single strand RNA, for it to replicate, it makes the positive strand, single strand RNA, and then uses this strand to make more of the genome, the negative single strand RNA. And the negative strand RNA is never translated or transcribed. Let's see, translated into protein. Only the positive strand RNA is translated into protein. All right, any question about that? If there's no questions, let's look at a picture. It's actually two pictures showing you the replication of a RNA virus, how the virus infects a cell and multiplies. We're going to look at this with a positive sense single-strand RNA virus, and then we'll look at it, the next slide, a negative sense strand RNA virus. If it's a double-strand RNA virus, you kind of have this going on. It's just that this is both strands of the RNA. Anyways, the virus attaches to the cell membrane, gets inside the cell. The RNA is released into the cytoplasm. This is a positive single-strand RNA, so that RNA can be used as messenger RNA to immediately be transcribed into viral proteins. The positive single-strand RNA will replicate, making negative strand RNA, and then the negative strand RNA is replicated to make more positive single-strand RNA, which is the genome of the virus. And then the genome of the virus will get together with the viral proteins right here, getting together, forming the new virions. And then the cell will release the viruses either in the lytic life cycle or by the budding life cycle. Any question about any of that? You don't need to know about the families. All right, so let's look at the replication of a negative strand, single-strand RNA virus. It too merges with the cell membrane, gets inside the cell. The virus is encoded, and then the RNA is in the cytoplasm. This RNA cannot be immediately read by the ribosome to make viral proteins. What happens is that virus makes the positive strand, which is now the darkly coded one, I think. You know, this is shown differently, so I'm not sure what's going on here. It looks like the dark one is the negative strand up here, but right here it's unclear what's going on. So anyways, the negative strand RNA makes the positive strand RNA, and then uses the positive strand RNA to make more negative strand. Maybe that's going from there to there. And then that's negative and that's negative. Any question about any of that? So that's how the negative strand multiplies its genome. And then the positive strand, corollary to the genome, is translated into the viral proteins, which we're not really seeing that here, maybe this slide here. So it's the dark ones are translated. And now the color changes. Oh well. Anyways, then the viral protein gets together with the negative strand genomic RNA of the virus, and then the virus assembles, and then the viruses are released either by the budding life cycle or by the lytic life cycle. Any question about RNA viral multiplication in animal cells? No questions? I'm going to move on. All right. Let's talk about the retroviruses. You need to know that the multiplication of the retroviruses, the family Retroviridae, is a little different than all other RNA viruses. And you will understand why once I begin talking about it. The retroviruses include the lentiviruses. These include HIV-1, HIV-2, which cause AIDS in humans, and another lentivirus is feline leukemia virus, which actually causes leukemia in cats, but the cat gets sicker and sicker, and it essentially acts like it has AIDS. But it's actually leukemia, which just makes the cat sicker and sicker. And the immune system of the cat gets very, very weak, and so that the kitty does essentially have no immune system. Retroviruses are unique in that they contain the gene for the enzyme reverse transcriptase. And reverse transcriptase is a protein which is an RNA-dependent DNA polymerase. What that means is that this protein can make DNA from an RNA template. And the word retrovirus comes from the first two letters of reverse and then the first two letters of transcriptase. So retrovirus. Any question about any of that? All right, the reverse transcriptase protein is actually in the virus. So if we look at a retrovirus, it is an envelope virus showing you there, and it has spikes on it. And there's the capsid there. I think. That actually looks like it has 3, so maybe that's the capsid there, the green one. Yeah, the green one's the capsid. So it looks like in this artist rendition, the yellow one and the blue one are both the envelope. And I'm not sure why there's 2 there. That should be 1. The virus has an RNA genome, of course, and it's a positive strand. What's a little unusual for this virus is it has two molecules of the positive strand RNA. And you don't need to know that specifically, so I'm just showing you the picture. But it has reverse transcriptase in there. Does anyone see it? Oh, there it is there, right there. Which you can barely see. Let me blow that up a little more. Oops, too far. Reverse transcriptase right there. And that means that the protein is transmitted with the virus, and as soon as the virus infects a cell, the reverse transcriptase can read the RNA to start making DNA. So attachment, penetration, and encoding of retroviruses are basically the same or similar to other animal viruses, but the biosynthesis stage is very different. And let me open up a window here. Not a window. A Microsoft Word document, which might not be the best, but it'll work. Move this over here. So, retroviruses have a positive strand, single strand RNA, and reverse transcriptase, which I'm just going to abbreviate RT, and that really should be on a different line, but we're going to just go with it. Can take that and make single strand DNA. And then the reverse transcriptase can take that, oops, wrong one, and make it double strand DNA, which I guess we should have DS. Any question about that? The double strand DNA then moves into the nucleus of the cell, and it integrates with the cell's DNA, so it integrates into one of the chromosomes of the cell, and then permanently becomes a part of the DNA of that cell. And then that viral DNA in the host DNA can be transcribed, making positive RNA, single strand RNA, which this strand can be used by the ribosome, so it moves out of the nucleus into the cytoplasm. And then the host cell ribosomes will read it and make the viral capsid proteins in protein translation. And then the positive single-strand RNA can move out of the, does that happen in the cytoplasm? Can move out of the nucleus and get together with the capsid proteins, making the new progeny virions. And then these progeny virions will come up to a membrane in the cell and bud out of it, releasing the new virion of the animal retrovirus. Any questions about retroviruses? So you can see what retroviruses are different. They're an RNA virus, but they replicate through a DNA intermediate. And this DNA intermediate goes into the nucleus and becomes a permanent fixture, meaning it recombines with the DNA of the host cell and permanently stays in there. But it can replicate, making a viral genome, the positive strand, which can be used as messenger RNA to make the viral protein, and then they get together for the progeny virions. In the human germline, we have about 5 retroviruses in our germline DNA. Fortunately, all of those retroviruses are defective and they cannot replicate. So the retroviruses, although they're carried in the human genome, the retroviruses are, what should I word? Dead viruses? Viruses unable to replicate? And they have become a portion of the human DNA. Mice are not so fortunate. They have a retrovirus in their genome, and that retrovirus can replicate and then be shed by the mice, infect other mice cells and do other things. And this retrovirus in mice increases their risk of getting cancer. And we're not going to talk much about that, but as you can imagine, any virus that replicates into DNA and inserts itself into DNA, well, it can mutate that DNA and then lead to cancer. And in the mice, the retrovirus does that. And that gene, not the gene, the virus is in the mouse genome. Okay? Fortunately, in humans, like I said, we have about 5 retroviruses in our genome, but they are not working. They're non-functional viruses, non-functional retroviruses. Any question about any of that? And I'm not going to ask you how many retroviruses do we have in our genome. That's just something for you to chalk up. It's I don't know, something that's good to know, but it's not terribly important. If there's no questions, let's look at the multiplication of retroviruses in this slide. A retrovirus binds to the cell membrane of a cell. Let's see if I can blow that up a little. It's brought into the cell, too far. Uncoded, and the reverse transcriptase immediately transcribes the positive strand RNA genome into DNA. And it does go through a single strand DNA, and then the single strand is made into a double strand DNA. That double strand DNA moves into the nucleus. Up here we have it here, sorry. So that's actually the DNA of the host cell chromosome. The viral RNA is made into viral DNA, and it does go through a single strand, so that should really be a 2-step. And the reverse transcriptase makes it double-stranded. The double-strand DNA then moves into the nucleus, and then integrates into the host cell's DNA, integrating into one of the host chromosomes. At this point, with the chromosome having a piece of viral DNA in it, remember we have the term provirus. I never mentioned that earlier with retroviruses, but that is the term that is used for a retrovirus also, when the viral DNA is inside of our DNA. That provirus can be transcribed, making positive strand RNA, which is the genome of the virus, but that is also the strand that's read by the ribosomes, and then that RNA will go into the cytoplasm, be translated into the viral proteins. The viral proteins, oops, one too far, will then get together with the virus, making the naked portion of the retrovirus, and then that retrovirus will push up against a membrane and then break it off as it buds out of the cell. Any question about retroviruses? And then that virus can infect another cell. All right, so that's retroviruses. Let's move on to coronaviruses. I'm talking about this one because we're in a current coronavirus epidemic. If you don't know about coronaviruses, they are single positive strand RNA viruses. They are enveloped. And here you can see coronaviruses. And if you notice, this picture looks familiar. This is the picture that I selected for microbiology a long time ago, before COVID-19 was ever around. And I just kept it because it was so appropriate. This is a coronavirus, and this is what they actually look like. Okay? Any question about that? Coronaviruses have been around for a long time, long before COVID-19. They are the second most common cause of the human colds. So many coronaviruses, several species, cause the human colds. And it is true that they are the second most common. The rhinoviruses of family Picornaviridae are the most common cause of the human cold. But these are the second most common cause of the human colds. They do cause a number of serious diseases, and COVID-19 is a species that causes serious disease. All of them, SARS, MERS, and COVID-19, are cause acute, severe respiratory problems in people. SARS is actually named after that, severe acute respiratory syndrome. COVID-19 is actually SARS-2, if you've never heard that name, and I'll go through it a little more in the next slide. COVID-19 also does that. And then MERS, Middle East Respiratory Syndrome. These are all three highly related viruses. And all of them can cause mortality in humans. Fortunately, COVID-19 has the least mortality of the three. MERS has the most mortality of the three, but a good thing about MERS, the highest mortality rate, about 36% of people who get MERS die. But the good thing about MERS is it doesn't spread from people to people. Most of the people who come down with MERS are in contact with camels a lot. So apparently MERS is a virus that camels have, and it can infect people from the camel. But once the people get MERS, they're unlikely to spread it to other people. Any question about that? SARS has a higher mortality rate. The mortality rate of SARS, they estimate, is around 10%. SARS happened in 2003. I think it happened one other time after 2003. And fortunately, the World Health Organization, in particular China, got it under control, and this virus may be extinct. Okay? Or it's in some animal reservoir and it hasn't hit the human population again. But like I said, it is a lethal virus. One good thing about SARS is, as far as we know, everybody who gets it is symptomatic, and that's why they were probably able to control it. COVID-19, if you don't know, there are a lot of asymptomatic people. People who are young, like in their low 20s and teenagers and children, about 50% of the patients are asymptomatic. They will not know that they have COVID-19. And when you're asymptomatic, you can spread it. And COVID-19, they were not able to control, and it quickly became worldwide. I haven't looked recently, but the last belief was the mortality, the last time I looked, which was about 2 months ago, so it might be out of date now, but the mortality rate, they estimate, is between 0.2 to 3%, meaning between 0.2 to 3% of people who get infected will die of COVID-19. The danger of COVID-19 is it spreads much more easily than the flu, and the older versions of the virus, if one person was infected, they were likely to infect 1.4 to 2.5 others, and that's why the virus numbers kept on expanding. The new version is even higher than this. I think there's 2 new versions. One comes from Africa. Does anybody remember where the other one comes from? There are 2 versions that are going around, and the world scientific community is worried about them, the medical community, because they don't.
The other one was in the UK.
In the UK, and the others in South Africa. Like I said, there's 2 new variants, and we don't know if the vaccine will work on these new variants. And these 2 new variants are much more likely to spread. So that when a person gets infected with the new variant, they will on average infect more than 1.4 to 2.5 people. So their infection rate is higher. Any question about any of that? Okay. And I have not read the latest update on the new variants, so I could be, I don't know, maybe a week out of date. But my understanding was the new variants hadn't been around long enough, so we weren't really sure if the vaccines were going to work on the new variants. So far, all of the vaccines work on all of the variations of COVID-19 that we know of, but the new variants, like I said, we don't know of. I think there was a Chinese-made vaccine that one of the variations of COVID-19, it got through the vaccine. I think on the South African vaccine, that the new, excuse me, not the South African vaccine, the South African variant, that it does get through at least one of the vaccines, meaning people can become infected, but when they become infected, they're much less likely to get severe symptoms from COVID-19. So the vaccine is giving some protection, but it's not giving total protection. And I don't remember which vaccine that was. But South Africa is using one of the vaccines, and they found that the new variant was getting through one of the vaccines. We'll just word it that way. All right, let's talk a little bit more about COVID-19. Its strict scientific name is severe acute respiratory syndrome coronavirus 2, and the abbreviation for that is SARS CoV covariant coronavirus 2. And this is so similar to SARS that not even the scientific community is calling it that, because it'll get confused with SARS. Most people are calling it COVID-19. COVID-19 stands for coronavirus, COVID, or COV, I guess, coronavirus. And then the ID-19 stands for identified in 2019. COVID-19 is closely related to bat coronaviruses. It likely originated from 2 bat coronaviruses and a pangolin virus. Pangolin viruses are spiny anteaters, and the majority of the coronavirus came from the 2 bat coronaviruses, that only a few genes encoding the spikes of this virus came from the pangolin. So it looks like COVID-19 is a recombination of 3 coronaviruses that probably, how do I say that, originated fairly recently, and also recently got into the human population. They think, although nobody really knows, that before spreading from some original animal, it got into a pangolin as an intermediate. A pangolin, once again, is a spiny anteater. And the pangolin may have introduced it to humans. It's thought that in an animal market in China, in the city of Wuhan, that the animal market may have had the COVID-19 in one of the animals, perhaps the spiny anteater, and then the virus infected a human who either bought the pangolin or was selling the pangolin, and then the human in Wuhan spread it to other humans. That is thought to be what's happened, but in reality we don't really know. And the Chinese are arguing that this virus may have been around long before and was just rare, and then it went to Wuhan, and for some reason it spread, especially in the market, the animal market in Wuhan. I can state that the Chinese have stated, we've had 2 or 3 instances where a lethal virus came out of these animal markets in China. SARS in 2003 was the first, and I think SARS the second time came out of an animal market, and then COVID-19 came out of an animal market. And the Chinese have come to the conclusion that having all these strange animals in their animal markets is a breeding ground for getting new viruses into the human population. And so they've decided to close down those animal markets and stop selling the really weird animals. Like, most people don't eat spiny anteaters. Okay? Any question about any of that?
So if this is true, that it came from a bat and a spiny anteater, and then somehow went into humans, so can humans still transmit that back to animals?
Yes, humans can transmit it back to animals. I know that there are several cases where cats and dogs have come down with COVID-19, and they got infected from their owners. I don't know if any humans spread it to bats or spiny anteaters, but since the virus seems like it grew in bats and spiny anteaters, probably they could. Any question about that? But yeah, humans can spread it to animals.
Okay, and is there, I'm drawing a blank, so I can't remember exactly what the word is for that, specifically transferring from animal to human and then back to animal.
Well, transferring from animal to human, they have the term zoonoses. Is that the term you're talking about?
Yeah.
Yeah, I don't know of a term for transferring from humans to animal. And if somebody knows it, you can give it. But actually, I don't know the term. Uh, I'm trying to think. Other than cats and dogs, I don't know of too many cases of humans transmitting viruses to animals. But as you know, sometimes when you come down with a cold or the flu, you can spread it to your dog or your cat. And they can spread it to you too. It tends to be that both colds and flus do not hit both humans and dogs and cats, but every once in a while you can get one which will spread that way. Colds and flus do tend to hit horses and horses and pigs and birds, and then spread to humans easily, and humans can spread them to those easily. But occasionally we can spread it to our cats and our dogs, and they can spread it to us too. But only occasionally. They seem to be a little different, and so most viruses that live in dogs and cats don't spread to humans, and the same with most human viruses, but there's a few that do spread. Did that make sense?
Yeah, it did. Thank you.
Okay, all right. And yes, it is known, I guess maybe I didn't mention it, that the COVID-19, they sequenced the virus, and they found that the sequence is related or closely related to two bat coronaviruses, and then a very short segment of COVID-19, just a few genes of COVID-19, is similar to the pangolin coronaviruses. And so we do know that COVID-19 is a recombination of three coronaviruses. We don't know when that recombination occurred, but it likely occurred, well, fairly recently, at least in evolutionary terms. So let's say within the last 20 years. And probably it was surviving in some animal, and only recently has infected humans. All right, let's talk a little bit more about COVID-19. Transmission is primarily via droplets from coughs or sneezes within a range of about 6 feet. It may spread further than 6 feet, and I think the highest they found is maybe 12 feet, but the virus doesn't spread well if you're more than 6 feet away from someone. It can spread also from indirect contact, meaning somebody sneezes. Let's say somebody sneezes on the store and they sneeze on, what's something you grab onto? A can of soup. And then later on somebody comes and they touch that can of soup. You can get it from indirect contact, as I stated. But it doesn't tend to spread that way, meaning coronavirus tends to spread directly from people to people. It can spread other than that, but it tends not to. I'm just going to word it that way. Most of the cases of coronavirus, people get it directly from other people, being within 6 feet of someone who had it. It's likely breathed in. It rarely appears to be picked up by the hands and then carried to the face. And that's kind of surprising because most colds and flus, we actually pick it up on our hands and then we inoculate a mucous membrane on our face. And that's how most colds and flus are transmitted. But coronavirus, most of them, not always, but most of the time you actually breathe it in. But you can pick it up from your hands and then inoculated into your face. The virus can survive for 3 days on plastic or a metal surface, but it can only survive for a day on cardboard or paper. So one thing you might do with your mail, and I always do this, is I always let it sit for a day before I open it. And I don't ever touch, I don't know, you maybe can see this. Can you see that? I never touch the plastic because the coronavirus can survive on the plastic longer than that, longer than a day. But I always let it sit for a day before I touch the mail. And I used to be very good about things in the store. I would buy them in the store and then either alcohol them down. If they're in a paper box, I would let them sit for a day before I touch it. Nowadays, I just leave it in my car for a few days and then bring it into the house after that. But I haven't been alcoholing my bananas or my oranges, something like that. I haven't been doing that. But I do let them sit for at least a day before I even move them into the house. Now, is that true? Obviously, on perishables, I do immediately bring them into the house and put them into the refrigerator or the freezer. So those I'm not really safe about. But all the other things, I try to let them sit. Any question about COVID-19 and how it spread? COVID-19 mostly gives flu-like symptoms if you have symptoms. That includes a runny nose. Although the runny nose and the sneezing are less common than the flu, the shortness of breath and the deaths are more common. The mortality rate increases with age, with ill health of the patient, meaning an ill patient is more likely to die than a healthy patient. Men are more susceptible, more likely to die than women. And then for reasons that have never really been explained, blacks and Hispanics, maybe some other minorities too, are more likely to die than whites. And this has been seen in other countries besides the United States, where blacks and Hispanics were more likely to die or more likely to get a serious disease than whites. And nobody's really explained why that's happening. Okay? They do know why the mortality rate is very low in children. It turns out that children have a very good innate immune system, which what I mean by that is they have a first line of defense as their immune system, and it's very good because their immune system has not been around very long, so they don't have a bunch of antibodies to a bunch of viruses. So their first line of their immune system is very strong, and what it does is it attacks the virus very quickly and frequently can get rid of the virus with a non-specific defense. And I'm not going to go into what that is, but it's the white blood cells and the interferon and other means the body has for fighting viruses. So children have a very good antiviral defense, which is non-specific, and that non-specific defense is killing off COVID-19 shortly after it infects children. Adults don't have that strong non-specific immune defense because we have a good immune system. We have a lot of antibodies to a lot of viruses. The trouble is COVID-19 is a new virus, and so unless you've been infected with it in the last year, you don't have any antibodies to COVID-19. Or I guess if you have the vaccine, you would have the antibodies as well. So that's why the mortality rate is younger in children, but as the child gets older and then gets older as an adult and older as an older, the mortality rate is increasing because the virus can mount a more and more severe reaction. And it takes time for the body, the human, to make an immune response against a brand new virus. That immune response won't happen for about 2 weeks. And by 2 weeks' time, COVID-19 can have already a severe infection. Any question about that? You don't need to know that. I'm not going to ask you why children have a lower mortality rate than young adults and much lower than middle-aged adults and much lower than elderly adults. But that's the reason why. The incubation period, this virus will infect a person, and the person will be 2 days to 14 days before they will have any symptoms. And that's assuming that they have symptoms. As I stated, some patients, about a third of all patients, 50% of all young people, and I'm talking about people, I don't know where the cutoff is, someplace in the 20s down to age, 0, about 50% of them will be asymptomatic. But if you have symptoms, you will be asymptomatic from 2 days to 14 days after you're first infected. And you can spread the disease, I think it's like day 3 or day 4. So after you're infected, 3 or 4 days after that, you can spread the disease even though you have no symptoms. I've already mentioned the estimated death rate for COVID-19 is 0.2 to 3%. The actual death rate, which this is 2 months old, was 2.1% of patients they know who became infected died in the United States. And these are only the people they know. So they have to know that they got infected and then know that they died of COVID-19. Prevention for COVID-19 is use correct hand washing. You should be washing your hand for at least 20 seconds with soap and water. You should keep your distance from other people. You want to stay 6 feet away from people, especially if they're not wearing a mask. If they're coughing, you should just get away from them, actually. Don't touch your own face until you washed your hand or use sanitizer. How good is sanitizer? Alcohol is very good at dissolving the lipid bilayer of COVID-19. That will destroy the virus. So sanitizers are very good at killing the virus. If you're sick, actually you should wear a mask even if you're not sick. But if you're sick with COVID-19, it's best to stay home, cough into your elbow or your tissue, and then use hand sanitizer or wash your hands after you cough. Any question about any of that?
I got a question about antibodies.
Uh-huh.
I heard that, if you suspect that you did have COVID-19 at the beginning, last year in March, around March, April, May, you can get tested for antibodies. Do that means that that your body created new,
Antibodies?
Antibodies to fight this virus, or?
Yes. However, last March was a year ago, and let's say somebody got infected last March, they probably made antibodies to COVID-19. But with time, those antibodies decrease. And after a year's time, this is almost a year, you might not have enough antibodies to give you immunity anymore. Okay? And they don't know how quickly the antibodies fall once you're infected with COVID-19. But you could, if you think you may have been infected with COVID-19, you could be tested. You'd have to go in probably to a special testing center. I don't know if this will be the general testing center or not, and ask, was I exposed to COVID-19? I would like to be tested. But after a year's time, your immunity may fall, and you could get infected again. There have been some people who have been infected at least 2 times. And there was one person, I read an article about it, they did get a serious disease 2 times from COVID-19. And he got it, I think, it was less than 6 months apart. Not everybody who gets infected with COVID-19 mounts an immune response, and they haven't quite explained that. Usually when you get infected, you will mount an immune response. But it seems like with COVID-19, there is a percentage of the people, and I don't know what that number is. It is a minority of the people, but some people don't seem to mount an immune response against COVID-19. These people should get the vaccine because if they're not mounting an immune response against COVID-19, that means they could get it multiple times. It turns out that the vaccine to COVID-19 probably will not be long-lasting. Like with polio virus, the vaccine lasts a long time. If it's the oral polio vaccine, that actually lasts nearly your lifetime. But even the dead polio vaccine, it lasts for at least 20 years. COVID-19 will not last, the vaccine will not last that long. They don't know how long it will last. I'm hoping it's going to last for at least 1 year, but you might need to get that vaccine a couple times in 1 year. And we will learn more about how long the vaccine will last with time. But they don't expect that it will last long. With the flu, as I mentioned, the vaccine lasts at best for 3 years. And COVID-19 probably will not last that long. This virus seems to change. And we're talking about the family. In COVID-19, as you know, there's a large number of variants now. The original virus that came from Wuhan is no longer circulating. It was replaced by a virus that came, I think, from northern Italy, someplace in Europe, and that virus is now being replaced by a virus that came from Europe, which will probably be replaced by either the UK virus or the South African virus. So this virus does seem to change fairly rapidly. All right, I've gone past the lecture time, so let me end this. If there's no further questions, we will continue here next time, and I will see you in the lab if you have questions at 6:30. If you don't have questions, you don't need to come to the lab. Okay? All right, goodbye.